Hello, and thank you for joining us today for another TissueGnostics webinar. My name is Carly Briffa, and today we're joined by Dr. Jiang from the Department for Hematology, Cell Therapy, and Homeostasology at the University Hospital of Leipzig to talk about the oncogenic role of the lysine-specific demethylase KDM1A in chronic lymphocytic leukemia.
Thanks for the invitation. I'm glad to present my project here. My name is Jiang. I'm going to talk about the oncogenic role of the lysine-specific demethylase KDM1A in chronic lymphocytic leukemia, short as CLL. One hallmark of CLL is the overexpression of TCL1 oncogene. The disturbance of epigenetic mechanisms is also involved in CLL pathogenesis. It has been reported that DNA methylation patterns varies among B cells from different developmental stages, and it predicts two major subtypes of CLL, namely IGHV mutated and unmutated. Studies have shown that aberrant DNA methylation can be detected before disease onset, and TCL1 might mediate transcriptional silencing through protein-protein interaction in murine CLL. However, the interplay around TCL1 and epigenetics in CLL is not fully understood. To address this, we performed a mass spectrometry analysis of TCL1 interactome in CLL patient samples. Via gene set overrepresentation analysis, we found different functionally annotated clusters interacting with TCL1, including epigenetic modifiers. This cluster we found interesting because epigenetic regulators are less well established in CLL. Here you can see that we have identified a panel of chromatin modifying enzymes. The histone modifier KDM1A has not been reported to interact with TCL1. KDM1A removes methyl groups from lysine 4 and 9 of histone 3, leading to transcription repression and activation respectively. Interestingly, it was shown to promote disease progression in acute myeloid leukemia. Nowadays, KDM1A inhibitors are being tested in clinical trials. Therefore, we would like to have a closer look of KDM1A in CLL. We hypothesized that KDM1A is involved in the pathogenesis of CLL, and we further ask, how does TCL1 impact KDM1A function or activity? What is the biological outcomes of increased KDM1A levels in CLL? Which KDM1A mediated pathways contribute to the pathogenesis of CLL? As a validation of mass spec results, we investigated the interaction of KDM1A with TCL1 in primary CLL cells, murine leukemic cells, and cell lines. Interestingly, they interacted in the nucleus, which we think is an important sign for TCL1 affecting the epigenetic function of KDM1A. Furthermore, we observed higher demethylase activity in TCL1 transgenic B cell line, and mass spectrometry of histone post-translational modifications identified differential histone markers in TCL1 negative and positive B cells. Lower methylation levels of H3K9 were observed in TCL1 positive cells. Based on this, we think that the overexpression of TCL1 in CLL might enhance the activity of KDM1A. As a second step, we would like to investigate if KDM1A is involved in CLL pathogenesis by testing its biological relevance. We could show that KDM1A is overexpressed in CLL compared to healthy B cells at protein levels. Analysis of gene expression profiling data of patients from the CLL-8 trial showing different pathways correlate with KDM1A levels. For example, DNA replication and BCR are positively correlated with high KDM1A levels, while cytokine receptor interaction is inversely correlated with high KDM1A levels. Using the differentially expressed genes in KDM1A high versus low group, here we identified 3 clusters. And the level of KDM1A are significantly higher in cluster 3 in comparison to cluster 1. Furthermore, patients in cluster 3 presented adverse prognostic features as higher white blood cell count, higher thymidine kinase serum levels, higher TCL1 levels, as well as higher rate of TP53 defects and shorter telomere length. Consistently, patients in cluster 3 have shorter progression-free survivals, the effect mainly derived from the FC trial arm. Altogether, higher KDM1A levels and the associated gene signature correlate with poor clinical outcome in CLL. Next, we planned to analyze the functional role of KDM1A in CLL mouse model. For this, we have used mice that ubiquitously express short hairpin RNAs against KDM1A in a doxycycline-inducible manner. These mice were intercrossed with CLL mouse strain named TCL1 transgenic mice. Treatment with doxfood for 2 weeks already led to an efficient lockdown of KDM1A in blood, spleen, and bone marrow. To assess the effect of KDM1A lockdown on blood parameters, we continuously treated mice with doxycycline food when the white blood cell counts were around 50,000 per microliter. As you can see here, in contrast to immune TCL1 transgenic mice, where the white blood cell counts rise with time, the white blood cell counts in KDM1A lockdown mice remain stable. The number of leukemic cells in peripheral blood stays also stable when KDM1A is locked down. Furthermore, we observed reductions of leukemic cells in spleen and bone marrow with short-term and long-term dox treatment. Quantification of immunofluorescent confirmed reduction of B-cell infiltration in spleen upon KDM1A lockdown. Additionally, lower proliferation rate shown by K67 staining and higher apoptotic rate of B cells were observed in the spleens of KDM1A lockdown mice. Besides the impact on leukemic cells, we would also like to know the effect of KDM1A lockdown on other cell compartments, for example, T cells, which have been reported to be skewed in immune TCL1 leukemic mice. Here we show a reduction of T cells in peripheral blood and spleen of leukemic mice with KDM1A lockdown. The decreased ratio of CD4 to CD8 cells in immune TCL1 mice was reversed upon KDM1A lockdown. And less T cells were observed in the splenic germinal center in KDM1A lockdown mice, shown by immunofluorescent staining. It is also known that KDM1A inhibition increases macrophages differential marker, as we could show an increase of CD11B, CD40, F4AT macrophages in peripheral blood and spleen. Quantification of immunofluorescence confirms that KDM1A lockdown leads to increased population of macrophages in spleen. However, when lockdown KDM1A in macrophages, their supporting effect on CLL cells was impaired, as less Ki67 positive proliferating CLL cells were observed in the co-culture when co-cultured with KDM1A lockdown THP1 cells. Moreover, KDM1A lockdown compromises the supporting effect of bone marrow stroma cells on CLL as well. We could say here in the co-culture system, lower cell viability was shown when lockdown KDM1A in leukemic cells or bone marrow stroma cells, or in both compartments in comparison to the wild-type condition. The support effect of HS5 stroma cells on CLL survival was impaired as well when KDM1A is depleted, as lower cell viability was shown in CLL cells co-cultured with KDM1A lockdown stroma cells. Together, these results indicate KDM1A-mediated impact is not only by its expression in CLL cells, but also to a relevant part through its presence in cells of the microenvironment. Consistently, the lockdown of KDM1A prolonged the median survival after DOX treatment by 30 days. I've showed that KDM1A lockdown reduces tumor burden and prolongs overall survival in leukemic mice. Next, we wanted to address KDM1A lockdown, how KDM1A lockdown reduces tumor burden on the molecular level. To this end, we have performed RNA sequencing of splenocytes isolated from leukemic mice with or without KDM1A lockdown. Principal component analysis shows distinct groups by genotype, meaning that KDM1A lockdown affects the gene expression. Around 1,000 genes are differentially expressed upon KDM1A lockdown. Because 81% of these genes are upregulated, we assume that KDM1A acts as a gene repressor in murine CLL. Gene set overrepresentation analysis identified p53 and apoptosis pathways are upregulated upon KDM1A lockdown. This is in line with our previous data that KDM1A depletion induces apoptosis and reduces tumor cells. GSEA confirms the enrichment of p53 and apoptosis pathways upon KDM1A lockdown. Those are the differentially expressed genes belonging to p53 and apoptosis pathways. For example, the pro-apoptotic marker MAPK9, Caspase-3, and JDD45 gamma are upregulated, supporting our hypothesis that KDM1A lockdown induces apoptosis. Next to a direct effect on genes mediating apoptosis, disrupting the interaction with the microenvironment can also drive CLL cells into death. We found several genes involved in cell adhesion and migration to be differentially expressed upon KDM1A lockdown. Three interesting molecules that are downregulated in this cluster are integrin beta-2, REBGAF3, and CXCR4, which are important for B-cell migration and infiltration. These results might give another hint that KDM1A lockdown can affect the microenvironment of CLL. As an epigenetic modifier, KDM1A can demethylate histone 3 on lysine 4, leads to transcriptional repression. To address this effect, assess the effect of KDM1A lockdown on histone modification, we performed chip sequencing in leukemic cells with or without KDM1A lockdown, confirmed by qPCR. Analysis of chip-seq data, we have identified more peaks in KDM1A lockdown splenocytes, meaning more H3K4 mesial 3 findings in KDM1A lockdown cells. Between the two groups, around 15,000 peaks were found unique in KDM1A lockdown groups. In 9-KEC pathway analysis of peak annotated genes revealed apoptosis pathway is enriched in cells from KDM1A lockdown mice. In addition, a total of 543 genes were both upregulated by KDM1A lockdown and associated with trimethyl H3K4 binding sites, of which 19 were transcription factors. Examples including apoptotic marker Caspase-3, MAPK9, transcription factor GATA1, GFI1B, and tumor suppressor TRIM16. Furthermore, analyzing the publicly available RNA-seq data and chip-seq data in primary CLL cells confirmed our findings in mouse model. The majority of the differentially expressed genes are upregulated in KDM1A low group. About 100 genes are both upregulated in KDM1A low cases and associated with H3K4 mesial 3 regions, and significantly more H3K4 meses3 peaks in KDM1A low group. Involved genes including caspase-3, MAPK9, and GFI1B. Together, this suggests that the transcriptional effect by KDM1A lockdown are also mediated secondarily through 2-methyl H3K4 regulated expression changes. To eventually validate KDM1A as a therapeutic target, we have tested different KDM1A inhibitors, which have been reported to work efficiently in other tumors. In primary cell, C12 and IMG were able to increase the methylation of histone 3 and induces apoptosis, showing by phosphorylation of p53 and cleavage of PARP. Furthermore, IMG in combination with BCL2 inhibitor venetoclax or with p53 activator idazalatinib presented high synergistic effect in primary CLL cells in inducing apoptosis. Analyzed per allicin-5 flow cytometry and Western blot for cleavage of PARP. Together, KDM1A inhibition alone or in combination are potential therapeutic strategy for CLL treatment. To sum up, first, I showed that TCL1 enhances KDM1A's activity. Higher KDM1A levels and the associated gene signatures correlate with shorter progression-free survival in CLL. From the in vivo study, KDM1A lockdown reduces leukemic cells, which is accompanied by upregulation of p53 and apoptosis pathways. Additionally, KDM1A lockdown reduces T cells and compromises the supporting effect of macrophages and stroma cells on CLL. Importantly, KDM1A lockdown prolongs survival in leukemic mice. And sequencing data suggests KDM1A as a transcriptional repressor in murine CLL. Pharmacological inhibition of KDM1A in CLL induces apoptosis and affects histone methylation. Altogether, we propose KDM1A as a pro-oncogenic marker in CLL cells and their microenvironment. This provides a rationale for targeting KDM1A in CLL. For this, I'd like to thank all my colleagues and our collaborators. And of course, thank you for your attention.